Good afternoon, dear students. Today we'll be discussing about histology of an epithelial tissue. Epithelium is an avascular tissue composed of sheets of cells that cover the exterior body surfaces and line internal closed cavities, including the vascular system and body tubes, such as the alimentary, respiratory, and genitourinary tracts, communicating with the exterior. Epithelium also forms the secretory portion of glands and their ducts. In addition, specialized epithelial cells function as receptors for the special senses, such as smell, taste, hearing, and vision. Therefore, all epithelia can be classified into three functional groups. They are covering, glandular, and sensory types. Epithelial tissue performs different functions. These functions are the following: protection, for example in skin and in esophagus; absorption, for example in tubules of kidney and in small and large intestines; transportation, for example the cilia-mediated transport in the respiratory organs and in endothelium lining our vessels; secretion of mucus, hormones, and proteins, for example in glands; exchange of gases, for example in alveoli of the lungs; lubrication between two surfaces, for example in mesothelium of the pleural, peritoneal, and pericardial cavities; sensation, for example in some sense organs; contractility, for example in some exocrine glands containing the myoepithelial cells. General features or characteristics of epithelial tissue are the following. Epithelia are continuous sheet-like cellular layers with little amount of intercellular substance, separating the underlying connective tissue from the external environment or environment of internal cavities. Epithelial cells are closely opposed and adhere to one another by means of specific cell-to-cell adhesion molecules that form specialized cell junctions. All epithelia rest on a basement membrane. Epithelia are avascular tissues because don't contain blood vessels. They are nourished by diffusion of substances from underlying capillaries of the loose connective tissue. Epithelial cells exhibit polarity. Epithelial tissues receive a rich supply of sensory nerve endings. Epithelia have a high ability to regeneration. Some epithelial cells display surface modifications such as microvilli cilia and cilia on their apical paths. This is the morphology classification of covering epithelia depending on the number of cell layers and the shape of superficial cells. According to this classification, all covering epithelia can be classified into simple, stratified, and special types. Simple epithelium contains only one layer of cells. It can be classified into squamous, cuboidal, and columnar types. Stratified epithelium contains more than one layer of cells and can be classified into keratinized squamous type and non-keratinized. The last one can be classified into squamous, cuboidal, and columnar according to the shape of its superficial cells. Special epithelia include pseudostratified and transitional types. The pseudostratified belongs to the simple, and the transitional type belongs to the stratified. Simple squamous epithelium is composed of a single layer of uniform flat cells which all rest on the basement membrane. Their nuclei appear flattened. This type of epithelium is found inlining the alveoli of the lung, inlining the vessels and heart chambers where it's called endothelium, inlining the serous body cavities such as pericardial, pleural, and peritoneal, and the serous coats of inner organs where it's called mesothelium. Inlining the thin segments of loop of Henle of the kidney, inlining the smallest excretory ducts of some glands, and inlining the posterior surface of the cornea. Simple cuboidal epithelium is composed of a single layer of uniform cuboidal cells, which all rest on the basement membrane. The nuclei of cells are centrally placed and spherical in shape. This type of epithelium is found inlining the most tubules of the kidney, inlining the thyroid follicles walls, inlining the excretory ducts of some glands, and in eyeball, where it produces the pigment epithelium of the retina and the subcapsular epithelium of the lens. Simple columnar epithelium is composed of a single layer of uniform columnar cells, which all rest on the basement membrane. The elongated ovoid nuclei are located in the basal part of the cell. This type of epithelium is found inlining the gallbladder, inlining the uterus and oviducts, inlining the ducts of some glands, and inlining the stomach and small and large intestines. Pseudostratified columnar epithelium is composed of a single layer of non-uniform cells, which all rest on the basement membrane. The cells of this epithelium are different shape and height, and their nuclei occupy varying positions. This type of epithelium is found inlining the respiratory tract and some parts of male and female reproductive tract. In pseudostratified epithelium, the most widespread type of cells is ciliated columnar-shaped cells, bearing motile cilia on their apical surfaces to provide the cilia-mediated transport. Except them, there are also goblet or mucus-secreting cells and basal cells. The last ones have capacity to mitotic activity to provide the regeneration of this type of epithelium. All stratified epithelia contain several layers of cells, but only the deepest layer of cells there is in contact with the basement membrane. There are three types of stratified non-keratinized epithelium according to the shape of cells in the superficial layer: squamous, cuboidal, and columnar. In our organism, a more distributing type of stratified non-keratinized epithelia is squamous. Stratified cuboidal and stratified columnar epithelia have a limited distribution and can be found in some large ducts of the exocrine glands. Stratified non-keratinized squamous epithelium consists of three cell layers. They are basal, intermediate, and superficial. The basal layer is represented by one row of the cuboidal or columnar-shaped basal cells resting on the basement membrane. The intermediate layer is represented by several rows of polyhedral-shaped cells. The superficial layer is represented by 2 to 3 rows of latent-shaped cells. This type of epithelium is found inlining the oral cavity organs, inlining the esophagus, inlining the vagina, inlining the two vocal cords of larynx, inlining the lower third of the anal canal, and in anterior epithelium of the cornea. Epithelial tissues are relatively labile structures whose cells are renewed continuously by mitotic activity and stem cell populations. In all types of stratified epithelia, stem cells and mitosis occur only within the basal layer in contact with the basement membrane. So the basal layer of stratified epithelia is also called the germinal layer. Stratified keratinized squamous epithelium is similar to the stratified non-keratinized squamous, except that surface cells are non-nucleated instead of nucleated and presentation of 5 cell layers instead of 3. Stratified keratinized squamous epithelium is found in the skin to produce skin epidermis. Epidermis of skin consists of 5 layers of epithelial cells. These layers are called stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. The stratum basale is one cell deep layer of the cuboidal or columnar-shaped basal cells resting on the basement membrane. The stratum spinosum is characterized by several rows of polyhedral-shaped cells. The stratum granulosum is characterized by 1 to 3 rows of latent cells containing the centrally located latent nuclei and cytoplasm peeling by basophilic cholesteinin keratohyalin granules. The stratum lucidum is highly refractal layer containing several rows of latent eosinophilic cells. This layer looks like a wavy translucent clear strip for microscopy. The upper stratum corneum is characterized by 15 to 20 layers of latent non-nucleated cells called corneocytes. Transitional epithelium or urothelium has specific morphological characteristics that allow it to distend. Transitional epithelium belongs to the stratified type because it consists of 3 cell layers: basal, intermediate, and surface or superficial. This type of epithelium is found inlining the kidney calyces and pelvises, inlining the ureter, inlining the urinary bladder, and inlining the proximal part of the urethra. The basal layer of transitional epithelium is represented by the smallest cuboidal cells resting on the basement membrane. The intermediate layer is represented by average-size polyhedral or pear-shaped cells. The surface layer is represented by the largest, sometimes binucleate dome-shaped cells called dome cells or umbrella cells. Surface umbrella cells change their shape in response to stretching and to relaxing. They become flattened when an organ is filled by the urine. Next, our question is called glands. Glands are composed of glandular epithelial tissue and can be classified as endocrine glands and exocrine glands according to how the secretory product leaves the gland. Exocrine glands have 2 parts: a secretory portion, responsible for the secretory process, and a duct system, responsible for releasing of the secrets into the lumen of an organ or directly onto the body surfaces. Endocrine glands are ductless glands, secreting their products called hormones into interstitial tissue or directly into the bloodstream. Both exocrine and endocrine glands are developed from covering epithelium in the fetus by cell proliferation and invasion into the underlying connective tissue. Exocrine glands remain connected with the surface epithelium by the tubular ducts. All endocrine glands lose the connection to their original epithelium and therefore lack ducts. Today will be discussed only about exocrine glands. There are different classifications of exocrine glands, and this is a classification of them by their product. On the basis of nature of secretory product, the exocrine glands can be classified into serous, mucous, mixed, and sebaceous types. The parotid gland, the pancreas, and sweat glands are examples of serous exocrine glands. The goblet cells of intestinal and respiratory epithelium and stomach epithelium are examples of mucous exocrine glands. The submandibular and sublingual glands and glands in the trachea and esophagus are examples of mixed or seromucous glands. And the sebaceous glands presenting in our skin are example of sebaceous type of glands. This is the classification of exocrine glands according to the mechanisms of secretion. All exocrine glands can be classified into merocrine, apocrine, and holocrine types. of secretion based on the pathway by which the secretory products are released from the cell. In miracrine secretion, the secretory product is released from the cell by exocytosis without the destruction of cell cytoplasm. Secretory cells of the parotid glands and pancreas are examples of miracrine types of secretion. In apocrine secretion, the secretory product is released together with a part of the apical cytoplasm of the secretory cells. Secretory cells of the mammary glands are example of apocrine type of secretion. In holocrine secretion, the secretory product is released by disintegration of the entire cell. Secretory cells of the sebaceous glands are example of holocrine type of secretion. This is the morphology classification of exocrine glands. All they are classified into multicellular and unicellular types. Unicellular glands are composed of only single cells. Their secretory products are released directly onto the surface of an epithelium, and goblet cells are examples of this type of glands. Multicellular glands consist of numbers of secretory cells arranged in different organizations. The multicellular glands can be further classified into several subtypes on the basis of different criteria. On the basis of ducts branching, all exocrine glands can be classified into simple, containing unbranched duct, and compound, containing branched duct. On the basis of secretory portions number, all exocrine glands can be classified into branched, containing several secretory portions, and unbranched, containing a single secretory portion. On the basis of shape of secretory portion, all exocrine glands can be classified into tubular, which have a secretory portion like a tube, alveolar or acina types, which have a secretory portion like a sac, and tubular alveolar types, which have both tubular and alveolar secretory portions. The secretory cells, also known as glandular epithelial cells, have well-developed synthetic apparatus and numerous secretory granules in their cytoplasm. Outer structure of secretory cells depends on the nature of secretion produced by them. There are three types of secretory cells. They are protein-secreting cells, mucus-secreting cells, and steroid-secreting cells. The protein-secreting cells, or serous cells, have well-developed rough endoplasmic reticulum and Golgi complexes, and are filled apically with axiphilically staining secretory granules in different stages of their maturation. Spherical nucleus disposes at basal part of serous cells. Acini of the pancreas and parotid salivary glands are composed of serous cells. Mucus-secreting cells, such as goblet cells, also have rough endoplasmic reticulum and Golgi complexes and are filled apically with mucinogen secretory granules, but these contain glycosylated proteins called mucins. Flattened nucleus disposes at basal part of mucus cell. When mucins are released from the cell, they become hydrated and form a layer of mucus. Steroid-secreting cells are characterized by the presence of extensive smooth endoplasmic reticulum and predominant mitochondria with vesicular cristae. Both these organelles take part in steroidogenesis. There are steroid-secreting cells in the cortex of adrenal gland and in male and female gonads. All large exocrine glands are inner organs, consisting of parenchyma, stoma, and duct system. The secretory cells of a gland constitute its parenchyma and perform the essential function of gland. Stoma is supporting fibrous connective tissue with blood vessels and nerves, forming a capsule surrounding the gland from outside, and connective tissue septa, penetrating from capsule into the organ to divide gland into lobules. Ducts of glands are classified into interlobular ducts lying within lobules and interlobular ducts lying between lobules within connective tissue septa. It was last question of this presentation. Thank you very much for your attention.